Good evening. This is Miss Dagenford, and we are going to continue our discussion on the immune system by starting to look at adaptive immunity. Now, this gets complicated pretty fast, so I put at the beginning of this section this nice overview of the adaptive immunity. So, over here on the left, we see the humoral mediated immune response, antibody mediated. And then on the right, we see the cell mediated immune response. Now, it is a really nice chart. You just have to follow it. You can see the first exposure, and this is what's going to be the antigen. We'll look at that in just a second. And what we're going to look at is how the antigen provokes a specific B cell and then how we can get the T cells provoked and active. Now the T cells immediately you can see they've got like a little extra step here, so we want to focus on that a little bit. And then both of them kind of need this activation by the T cell as well. Kind of this provocation, the helper T cell gets them going. And then once we've got the first exposure, the second exposure really is the same thing, but we're using memory T cells and B cells. So we'll come back and look at this on occasion, but that will get you started. You can kind of look at this, pause the video, and maybe get a little glimpse of what's going to happen. Now, first, we need to establish why these are now specific. We saw with the non-specific that they just acted on molecules that a lot of viruses have or a lot of bacterial cells have. It's not that they were looking at a type of bacteria, but a lot of species of bacteria might have the same compound. And so thus it was non-specific. But here we've got the specificity. And the specific nature of this comes from receptors. Now, we're going to see themes that we've seen before. This protein shape is very important. Now, B cells have a receptor that's found in the membrane, and some of you will recognize this. It resembles an antibody. What they have is a constant. C is the constant region. The V are variable regions. And the variable regions, just as the name implies, that's going to give a specific shape to what's called the antigen binding site. So up here, I have drawn a couple to just indicate how they're different. And so you can see the measles, the antigen binding sites are going to look a little different than perhaps an influenza B. Or, take your pick, mumps, the common cold, flu, a certain kind of bacterial toxin. They are shaped differently because of that. Now, because we've been looking at this all year long, you should be able to kind of answer this question. How do we get this variability in a protein? So give it a pause and see if you can think through. Remember how do we make different kinds of proteins? Well, hopefully you are thinking back to the idea that these proteins are governed by genes. And so the variable regions are coded by separate kinds of genes, and we'll talk about that in a second. The constant regions, well, are constant. Those genes don't have much variation. And then you throw in mutations, and we can get some more variations from that. The transmembrane region just is a nice connection. Remember, these amino acids must be nonpolar in order to kind of go through those nonpolar tails of the membrane. Now, over here, you can see the fact that the receptor is going to interact with these antigens, and the antigens are found on the pathogen. The pathogen would be things like bacteria, viruses, fungal cells, anything that would be foreign, even a transplanted organ. We worry about that as well. So, fungal cells, they all have different antigens on their surface. And even the antigens can have slight different variations, and we call those epitopes. So, even an antigen could have slight variations that two types of antibodies or receptors might react to. So you see down here, antibody A or receptor A would focus on this epitope, antibody B would focus on that epitope, but antibody C doesn't really attach to anything. So those would be the ones that would react. Now, T cells have a somewhat similar arrangement, but they don't have that Y shape. T cells are just one long length, but you'll notice some similarities. Variable regions, the antigen binding site. So this is binding to an antigen. Now it's not going to directly bind to an antigen just floating around. So that's going to be an important difference. We'll talk about that in just a second. Now, we have, just go back just a second, we have close to 1 million, there it's been estimated, B cells. And then down here, it's been estimated that the T cells are even more, 10 million. 10 million different kinds of T cells. Now, if you made the connection between protein and genes, we have to ask the question, do we have, therefore, 10 million genes? Well, we don't. So that's kind of weird. How do we get 10 million variations of a T cell, but we don't have 10 million genes? Well, hang on. We'll answer that in just a second. Now, the T cells, they cannot or they will not be activated by an antigen displayed by a bacterial or a virus. They interact specifically to what's called an antigen presenting cell. So here is our antigen presenting cell, APC, and just as the name implies, an antigen presenting cell presents an antigen. And so you can see it's going to present this little particle, this antigen, on the surface of what's called an MHC molecule. We'll tackle that in just a second. So the MHC molecule holds it out, and what's going to happen is the proper T cell that has the right match is going to connect, and that's going to set off a signaling pathway. So the only thing we need to kind of answer is how in the world is this antigen presenting cell getting that antigen? And the answer lies in what's happening over here. Notice it's actually consuming, it's engulfing the pathogen, and then it's going to break it down and then hook these two up together, and it's going to go to the cell surface. Now, um, a quick view, instead of a rectangle and a triangle, what we actually see are the secondary and tertiary structures of this MHC molecule. So here's our MHC in the green. Now, your copy is somewhat harder to see. Feel free to color code it. This is going to be an antigen that is going to match that little groove. It's going to fit in that little groove. And in this case, the antigen presenting cell is actually signaling to a T cell. These are, think about the sentries or the guards, they go around gobbling up these antigens and then they present them. So they're going back to the soldiers and trying to find the one that matches. It's presenting the antigen to whatever T cell matches. So if this is a measles virus, just as an example, what it's trying to find is the T cell that matches the measles, that has the receptor that matches that measles antigen. And then therefore that turns on the T cell. We'll practice that in a bit. Now, let's go back to those genes questions. Um, what we end up having, I'm going to have to do this a little out of order here. What we end up having is a way to recombine these things. So we've got a generation of these receptors. Now, it's kind of crazy, and this happens with T cells as well. We've got the DNA of undifferentiated B cells. So we've got these immature B cells that are in a fetal system. They're being developed, and this is all the same gene. We've got 20,000 genes, but of those 20,000 genes, each subset can be recombined in some way. So what we see is a little bit of math here. So let's take a look at this. There are 20,000 genes. 1 million B cells are different, 10 million T cells that are different. How is this happening? Well, let's take a look at the gene for the light chain only, and you'll see that this gets complicated quite fast. We have 40 possibilities of 40 different variations in the variable region. And we have 5 joining segments. So if I go back to this picture, we have these variable regions. And then we've got these joining regions. And then we have an intron, and then we've got one constant region. Well, if you do the math, now this is the same kind of math if you've ever encountered a problem where they say things like, they went into the Baskin Robbins for the 31 flavors and they got 5 scoops of ice cream. Well, if there are 31 flavors, what are the possible combinations of ice cream 5 scoops that they could have gotten? Well, it's the same idea. You would multiply the possibilities. So you can have 200 possible variations. And so with these immature B cells, there's an enzyme called recombinase, and it will take these undifferentiated B cells and snip out the variations that it will cut. And this is a random process, and so you can get those 1 million B cells and 10 million T cells. So notice now this is the functional gene here. We have a variable region, a joining region, and a constant. If this were a different B cell, maybe you would have gotten this variable region and J1 for the joining. So it really is just a mix and match type of thing. And so this is the RNA, and just as we've learned before, the immature mRNA then needs to be processed. So we've got our 5' cap, the variable region, the joining region, and the constant region, that poly A tail that then goes out to our cytoplasm, and then we can get our variable region polypeptide, our constant region, and then lo and behold, we're going to get our receptor. And so every B cell that is developing will go through this process. It's one of the interesting things that we know that all cells in our body have the same kind of DNA, all of these chromosomes. Well, in our B cells, they actually don't. They will make different versions of the receptor genes, and then they become permanently changed. Once that recombinase snips out the different types and combines them, that B cell now becomes differentiated. And you get a B cell for that measles or mumps or the common cold or the flu. Now, something similar happens to this T cell, and in addition, it can also happen to the heavy chain. The heavy chain will give even more combination. The light chain is 200, the heavy chain is even more so. So it's been estimated that if you put all of these combinations together, you can get an astounding, just for the B cells, you can get an astounding 3.5 times 10 to the 6 numbers of these receptors. So that's going to give us kind of a nice beginning of all of these variations of these cells. So hope that helps get the conversation started. Um, and we will come back on Tuesday to talk about this.